Our final metabolic pathway then is photosynthesis. So when we're looking at photosynthesis, we're thinking of those organisms that produce the foods that other organisms eat. So things that we eat. Plants, algae, and multiple bacteria are photosynthesizers. The basic reaction is opposite of aerobic respiration. So in aerobic respiration, we started with glucose and oxygen, and we produced carbon dioxide and water and energy. In this case, we're going to start with carbon dioxide and some type, typically we think of water, H2O, but it could be something else like H2S, hydrogen sulfide. That is going to be used as the electron donor and proton donor. And we're going to produce a carbohydrate and some waste product. In this case, it could be sulfur as a waste product, or if it's water that we're looking at, then it'd be oxygen as our waste product, okay? There are two basic reactions that are going to occur. We're going to have light reactions where we're producing energy, or we're not producing, we're capturing energy and producing a form of energy that our cells can use, ATP. And then we have the light independent or dark reactions where we take the energy as well as the molecules that we've produced in the light dependent reactions, and we produce our carbohydrate. So depending on the type of organism that we're looking at, photosynthesis occurs in a lot of different areas. In plants and algae, photosynthesis occurs in the chloroplast, where you have the light dependent reactions in the thylakoid and then the light independent reactions in the stroma. In cyanobacteria, they have thylakoids, they just don't have the chloroplast. So they have the light dependent reactions within that thylakoid membrane still, and then they have the light independent reactions outside of that thylakoid membrane. In purple and green anoxygenic photosynthesizers, they're going to have a different area. So within the plasma membrane, where they're going to have their pigment containing molecules, and they're going to be able to trap light and go through the light dependent and light independent reactions. With anoxygenic organisms, they're going to have to use a lot more energy to produce the same amount of carbohydrates. Okay. So let's look at the light dependent reactions first, because those are typically the first that we see. Light dependent reactions, there are two different photosystems. If we're looking at aerobic first, they're using photosystems 2 and 1. They're named for how they were found, not how they are used. So in non-cyclic photophosphorylation, I'm going to go to the next slide. You can read through this, okay? In non-cyclic photophosphorylation, you're going to have water taken in by the organism. Water is going to be oxidized. The electrons are going to move into the reaction center of photosystem 2. Sunlight energy comes in and is taken in by the chlorophyll molecule and is going to activate the electrons. The electrons are going to jump into an electron carrier or transport chain. This helps to pump hydrogens across the membrane so that we have a high energy gradient again. The electrons move down energy gradients and eventually will enter photosystem 1. The hydrogens will flow through the protein ATP synthase, producing ATP. Now we have the electrons from the splitting of water over here in reaction center of photosystem 1. More sunlight energy comes in and is trapped, activates that electron again, or those electrons again, and they jump, and this time they're going to attach to NADP. And the hydrogen will also attach to NADP and will produce NADPH, which is what we need to produce our carbohydrate. So that's non-cyclic. Cyclic photophosphorylation produces a lot more energy. So remember, in like the purple and green anoxygenic photosynthesizers, they need more energy to produce the same amount of carbohydrate. So they use a cyclic photophosphorylation as well. Cyclic photophosphorylation allows us to use the same electrons over and over, and they just, as they get excited, they jump into an electron transport chain, producing more energy through the movement of hydrogen through the ATP synthase protein. But there is no reducing power, no NADPH produced. And we're not splitting more molecules, like more water, to produce more electrons. That same electron just moves back into photosystem 1. It gets excited again and then goes back over, moves back into photosystem 1, gets excited again. So this is why it's called cyclic photophosphorylation. This is not working, so I'm going up here, okay? Maybe. There we go.
In bacterial photosynthesis, a single photosystem is involved. When an electron is energized by absorption of light, it is ejected from the photosystem reaction center. The electron then passes down through an electron transport system and finally back to the reaction center. The energy released during this electron transport is used to produce ATP. Since the excited electron returns to the reaction center, this mechanism for making ATP is called cyclic photophosphorylation. No reducing power needed for biosynthesis is generated in this process. Plants and cyanobacteria utilize two photosystems which work sequentially to produce both energy and reducing power. First, a photon of light ejects a high-energy electron from photosystem 2. The electron lost from photosystem 2 does not return to photosystem 2, but is replaced by an electron generated from the enzymatic splitting of water and the release of oxygen. The electron then travels from the excited reaction center of photosystem 2 down an electron transport chain and finally to the reaction center of photosystem 1. This electron transport system generates a proton motive force that is used to produce ATP. Since the excited electron does not return to photosystem 2, this mechanism for making ATP is called non-cyclic photophosphorylation. When photosystem 1 absorbs a photon of light, it ejects a high-energy electron, which is used to drive the formation of reducing power in the form of NADPH. The ejected electron is replaced by an electron from photosystem 2.
Okay. And here you can see the photosynthetic electron transport chain and ATP synthesis.
In plants, photosynthesis occurs in specialized organelles called chloroplasts. The internal membranes of chloroplasts are organized into sacs called thylakoids. Surrounding the thylakoid membrane system is a semi-liquid substance called stroma. photosynthetic pigments are clustered together to form photosystems. When a photon of light strikes the reaction center of photosystem 2, it excites an electron. Two water molecules bind to an enzyme at the reaction center. This enzyme splits the water and uses the electrons from the water to replace the electrons removed from the reaction center. Oxygen is produced in this process. The primary electron acceptor for the light-energized electrons leaving photosystem 2 is plastoquinone. The reduced plastoquinone passes the excited electrons to a proton pump embedded in the membrane called the B6F complex. Arrival of the energetic electrons causes the B6F complex to pump protons from the stroma into the thylakoid space, thereby generating a proton gradient across the membrane. Because the thylakoid membrane is impermeable to protons, the protons in the stroma must pass through the channels provided by ATP synthase. As protons pass through, ADP is phosphorylated to ATP and released into the stroma. This process for making ATP is referred to as photophosphorylation. When photosystem 1 absorbs a photon of light, its reaction center passes high-energy electrons to ferredoxin. The enzyme NADP reductase then transfers the electrons to NADP to form NADPH. Electrons lost from photosystem 1 are replaced by electrons generated from photosystem 2. A small protein called plastocyanin, PC, then carries the electrons from the B6F complex to photosystem 1.
All right. So in anoxygenic or anaerobic organisms, they're not going to use water as their molecule that they split to gain electrons and hydrogens. They're going to use something else. Typically, they might use hydrogen sulfide, they might use hydrogen gas, they might use nitrite, anything that they can gain hydrogens from, but because they're using these other molecules that are harder to split, they need more energy. And so they're going to use just the light, or they're just going to use the single photosystem to produce as much energy as they can to split the molecule, and then they're going to use the hydrogens and the energy to produce their own carbohydrates. So now we're going to move into the carbon fixation step, or the light-independent reactions. All of the materials that were produced in the light-dependent reactions are going to then be used in the light-independent reactions to produce your carbohydrate. Typically, when we do this, the mechanism we use is called the Calvin cycle. And with Calvin cycle, we're going to take in carbon dioxide and we're going to bind it to another molecule called RUBP. And then we're going to reduce it to produce our carbohydrate, okay? So here's a very simplistic picture. I found this on Google Images. Here you have 3 carbon dioxides moving in. So each of the carbon dioxides that moves in will produce a 6-carbon molecule because we have a 5-carbon RUBP molecule there. And so that's a 5-carbon molecule. Carbon dioxide binds to it, producing a 6-carbon unstable molecule. This unstable molecule is going to immediately break into 2 3-carbon phosphoglycerate molecules. These 3-carbon phosphoglycerate molecules are then going to be reduced to glyceraldehyde 3-phosphate. And So with 3 carbons moving in, we produce 6 of these. That means we're going to have 6, we're going to produce 3 of these 6 carbon molecules, which means we have 6 3-carbon phosphoglycerates, which means we have 6 3-carbon glyceraldehyde 3-phosphates. We only get to gain 1 of these though, because this is a 3-carbon molecule and we're taking in only 3 carbons up here. So it takes 6 turns of the Calvin cycle to produce 1 glucose molecule. For every carbon dioxide that comes in, so for every 3 carbon dioxides that come in, we're going to produce 1 glyceraldehyde 3-phosphate. The rest of the glyceraldehyde 3-phosphates have to be used to regenerate the RUBP molecule. This is your figure showing the exact same thing. Here we have carbon dioxide coming in, producing these 6 carbon intermediates that immediately will break down into 3 phosphoglycerates. So we have 12 3-carbon phosphoglycerates. They're going to be reduced to the glyceraldehyde 3-phosphate molecules. We will gain from this 2 of the 3-carbon phosphoglycerates that can then be used to produce glucose or other molecules. The rest will be used to produce the 5-carbon RUBP molecules. And, yep, oops, sorry. There we go. I'm going to watch this, and then that's the end, okay, of this chapter.
The Calvin cycle is a series of reactions that results in conversion of carbon dioxide into the organic molecules needed to build new cells. It occurs in the stroma, which is the area of the chloroplast surrounding the thylakoid membrane. During the Calvin cycle, carbon dioxide, CO2, is added to a 5-carbon molecule called RUBP. The resulting 6-carbon molecule is unstable and quickly splits into 2 3-carbon molecules called 3-phosphoglycerate. Using energy from ATP and reducing power from NADPH, which are products of the light reactions, the pair of 3-phosphoglycerates move through a series of reactions and are converted into 2 molecules of glyceraldehyde 3-phosphate. When several of these glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle. To generate an entire new glucose molecule, the cycle has to turn several times, because each turn of the cycle adds only 1 carbon atom from each molecule of carbon dioxide.
And, all right, so I'm done with this video. I will have this posted shortly. Have a good day.